What if I told you that sitting in your kitchen right now, in your refrigerator, in your pantry, possibly even in the cup of tea you're drinking at this very moment, there are compounds so powerful, so remarkably intelligent in how they interact with the human body, that they can literally walk up to a cancer cell, cut off its food supply, and force it to die. And at the same time, those very same compounds can go into the nucleus of your healthy cells, find damage in your DNA, the kind of damage that quietly builds up every single day, and help repair it before it ever becomes something dangerous. I know that sounds almost too good to be true. I know it sounds like the kind of headline that gets thrown around on the internet to sell supplements, but I am not selling you anything today. I am Dr. Tasuku Honjo, and what I am about to share with you is grounded in peer-reviewed science, in cellular biology, in years of research that the mainstream conversation has been incredibly slow to catch up with. So stay with me, because by the end of this video, the way you look at your grocery list is going to be fundamentally changed. Let's start with something that most people don't fully appreciate, and that is just how fragile and yet how resilient your DNA actually is. Every single cell in your body contains about 3 billion base pairs of DNA, and every single day, each of those cells sustains somewhere between 10,000 and 100,000 instances of DNA damage. That is not a typo, that is not an exaggeration, that is the scientific consensus. Now, before you panic, your body has extraordinary repair mechanisms built in, enzymes and proteins whose sole job is to patrol your genome, find breaks and errors, and fix them. But here's the problem. Those repair mechanisms are not infallible. They get overwhelmed. They slow down as we age. They get disrupted by chronic inflammation, by poor sleep, by environmental toxins, by a diet that is chronically deficient in the very micronutrients they need to function. And when that repair process starts to fail, when errors accumulate faster than they can be corrected, that is when the door opens to cancer, to accelerated aging, to neurodegenerative disease, to all of the things we are desperately trying to avoid. So the question I want you to sit with is this: what if you could feed your DNA repair machinery? What if specific foods contain specific molecules that not only provide the raw materials your body needs to fix damaged DNA, but actively signal your cells to upregulate those repair pathways? Because that is exactly what the science is showing us. And the food at the center of this conversation, the one that keeps showing up in study after study, is one you almost certainly have access to right now. I want to talk about cruciferous vegetables: broccoli, cauliflower, Brussels sprouts, kale, bok choy, arugula, cabbage, and their relatives. Now I know, I can already hear some of you sighing. You've heard this before. Eat your vegetables. But I want to go much, much deeper than that today, because what is happening inside these plants at a molecular level is genuinely extraordinary, and most people have absolutely no idea. When cruciferous vegetables are chopped, chewed, or damaged, an enzyme called myrosinase comes into contact with compounds called glucosinolates. This contact triggers a chemical reaction that produces a molecule called sulforaphane. And sulforaphane is, without exaggeration, one of the most studied, most potent, naturally occurring compounds ever identified in the context of cancer biology and cellular repair. Let me tell you why. Sulforaphane is what is known as an NRF2 activator. NRF2 is a transcription factor, essentially a master switch inside your cells that controls the expression of over 200 genes. And the genes it controls are the ones responsible for producing your body's most powerful antioxidant and detoxification enzymes. We're talking about glutathione, superoxide dismutase, catalase, heme-oxygenase. These are the compounds your cells use to neutralize the reactive oxygen species, the free radicals, that cause DNA damage in the first place. When sulforaphane activates NRF2, it is not just neutralizing damage that has already occurred. It is fundamentally upgrading your cell's capacity to prevent damage going forward. It is like hiring 10,000 more security guards and giving each of them a better toolkit. But that is only the beginning, because sulforaphane does something else that is perhaps even more significant in the context of cancer. It inhibits histone deacetylases, or HDACs. Now, stay with me here because this is important. Your DNA is wrapped around proteins called histones, and the chemical modifications to those histones, whether they are acetylated or deacetylated, determine which genes get expressed and which ones stay silent. Cancer cells are notorious for using HDAC activity to silence tumor suppressor genes. Genes that would normally say stop dividing, this cell is out of control, get chemically switched off. Sulforaphane inhibits that process. It essentially reactivates those silenced tumor suppressor genes, forcing cancer cells to confront their own runaway growth. There is a landmark study from Johns Hopkins, the same institution where sulforaphane was first isolated, showing that sulforaphane could selectively target and kill cancer stem cells. Not just cancer cells in general, but the particularly stubborn, particularly dangerous cells that are responsible for tumor regrowth after conventional treatment. The mechanism involves disrupting the WNT signaling pathway, which cancer stem cells depend on for their survival. And critically, the research showed that sulforaphane was doing this while leaving healthy cells essentially unharmed. That selectivity, that ability to distinguish between a cancer cell and a normal cell, is precisely what makes this compound so remarkable from a pharmacological standpoint. Now, I want to talk about another food that deserves far more attention than it typically gets in these conversations, and that is green tea, specifically the polyphenol EGCG, epigallocatechin galleta. If sulforaphane is a master regulator of cellular defense, EGCG is a multitasking molecular ninja. And I say that not to be dramatic, but because the breadth of its mechanisms is genuinely difficult to summarize concisely. EGCG has been shown to directly inhibit DNA methyltransferases, the enzymes responsible for methylating DNA, which is another epigenetic mechanism that cancer cells exploit to silence tumor suppressor genes. In multiple cancer cell lines, treatment with EGCG resulted in the reactivation of genes like P16, ARR-beta, MGMT, and HMLH1, all of which play critical roles in cell cycle regulation and DNA mismatch repair. In plain English, EGCG was helping cells remember how to protect themselves against cancer. But EGCG also does something on the structural level of DNA repair. Research published in the journal Carcinogenesis demonstrated that EGCG enhances the activity of key DNA repair enzymes, specifically in the context of oxidative DNA damage. It appears to increase the expression of genes involved in base excision repair, one of the primary pathways your body uses to fix the kind of DNA damage that accumulates from normal metabolic processes and environmental exposures every single day. So when you drink green tea, you are not just getting antioxidants that scavenge free radicals. You are actively supporting the molecular machinery that fixes your genome. And then there is the question of angiogenesis. This is one of the most critical vulnerabilities of cancer, and it is one that EGCG exploits brilliantly. Angiogenesis is the process by which tumors grow new blood vessels to feed themselves. Without a blood supply, a tumor cannot grow beyond a few millimeters. It literally starves. EGCG has been shown in both in vitro and in vivo studies to inhibit VEGF, vascular endothelial growth factor, which is the primary signal tumors use to recruit those new blood vessels. By blocking VEGF signaling, EGCG essentially tells a growing tumor, your supply chain is cut. This is not a theoretical mechanism. This is a well-documented, reproducible, peer-reviewed finding that has been replicated across multiple research groups on multiple continents. Now let us talk about something that connects everything I have been describing, and that is chronic inflammation. Because here's the thing that most people don't realize. Inflammation is not just a symptom of disease. In many cases, it is a driver of DNA damage. The same reactive oxygen and nitrogen species that are generated during inflammatory processes are directly genotoxic, meaning they damage DNA. Chronic, low-grade systemic inflammation is essentially a slow, continuous assault on your genome. And the modern Western diet, high in refined carbohydrates, industrial seed oils, processed meats, and low in fiber and phytonutrients, is profoundly pro-inflammatory. It is constantly feeding that fire. This is where turmeric, specifically its active compound curcumin, enters the picture. Curcumin is arguably the most well-studied natural anti-inflammatory compound in existence. It works by inhibiting NF-κB, which is arguably the most important pro-inflammatory transcription factor in human biology. NF-κB controls the expression of cytokines like TNF-alpha, IL-6, and IL-1 beta, the very molecules that sustain chronic inflammation. By blocking NF-κB, curcumin is not just reducing inflammation as a symptom, it is interrupting the upstream signaling that drives the inflammatory cascade in the first place, and curcumin has direct effects on DNA repair as well. Studies have demonstrated that curcumin modulates the expression of p53, often called the guardian of the genome, which is a tumor suppressor protein that coordinates the cellular response to DNA damage. When DNA is damaged, p53 either triggers repair or, if the damage is too severe, instructs the cell to undergo apoptosis, programmed cell death. This is an essential safety mechanism. Cells with irreparably damaged DNA should die. Cancer is, in many ways, the failure of that mechanism. Curcumin appears to help restore the integrity of that p53-mediated response, making cells both better at repairing damage and better at eliminating themselves when repair is not possible. One important note about curcumin: its bioavailability in isolation is actually quite poor. Your digestive system processes it quickly before it can reach systemic circulation in meaningful concentrations. However, research has consistently shown that combining curcumin with piperine, the active compound in black pepper, increases its bioavailability by up to 2,000%. So when you are cooking with turmeric, add black pepper. Always. This is not a small detail. It is the difference between consuming curcumin as a flavor and consuming it as a genuinely therapeutic compound. I also want to spend a moment on berries, specifically dark berries like blueberries, blackberries, and pomegranate. These fruits are extraordinarily rich in anthocyanins and ellagic acid, which have demonstrated remarkable capacity to modulate DNA repair pathways and induce apoptosis in cancer cells. Ellagic acid in particular has been shown to inhibit the activation of carcinogens, compounds that become DNA damaging only after they have been metabolically activated in the body. By blocking that activation step, ellagic acid is essentially intercepting the damage before it even begins. Pomegranate deserves special mention because it contains urolithins, compounds produced when gut bacteria metabolize ellagitannins from the fruit. Urolithins have been shown to activate mitophagy, which is the selective autophagy of damaged mitochondria. This matters enormously for cancer prevention because dysfunctional mitochondria are a major source of the reactive oxygen species that damage DNA. By clearing out damaged mitochondria and replacing them with healthy ones, urolithins are addressing the problem at its metabolic source. And let us not overlook the importance of folate, found in abundance in dark leafy greens, legumes, and avocados. Folate is not just important for pregnant women, as the public health messaging has historically focused on. Folate is essential for DNA synthesis and repair across the entire lifespan. It is a critical cofactor in the methylation cycle, which governs both the production of new DNA bases and the epigenetic regulation of gene expression. Folate deficiency creates uracil misincorporation into DNA, essentially inserting the wrong building block into your genome, which leads to strand breaks that must then be repaired. When folate is chronically insufficient, that repair burden increases significantly, and the risk of mutations accumulates over time. Now I want to bring this all together with something practical, because information without application is just trivia. The science I have described today is not telling you to take a supplement. It is not telling you to drink a particular branded juice or follow some extreme protocol. It is telling you something far simpler and far more powerful. The foods that your biology evolved alongside, the foods that have been present in human diets for tens of thousands of years, contain molecular signals that your cells are literally designed to respond to. When you eat broccoli, your cells upregulate their antioxidant defenses. When you drink green tea, your DNA repair enzymes become more active. When you eat turmeric with black pepper, you suppress the inflammatory signaling that drives genomic instability. When you eat pomegranate and berries, you are feeding your gut microbiome the precursors it needs to produce compounds that clean up your mitochondria. This is not magic. This is biochemistry. And the most important takeaway is not that any single food will cure or prevent cancer. The research does not support that kind of simplistic framing, and I would never want to mislead you in that direction. What the research does support, compellingly, consistently, and across decades of investigation, is that a dietary pattern rich in these phytonutrient-dense foods creates a fundamentally different cellular environment than one that is devoid of them. An environment that is more resilient to DNA damage, more capable of repairing errors before they become mutations, and far more hostile to the conditions under which cancer cells thrive. If there is one thing I want you to take from this video, it is this. Your genome is not a fixed, static thing waiting passively to develop disease. It is a dynamic, responsive system that is in constant conversation with your environment, and the food you eat is one of the most powerful inputs into that conversation. Every meal is an opportunity to send your cells a message. The question is simply, what message are you sending? I hope this has been valuable for you. If it was, please share it with someone you care about, because this is the kind of science that deserves to reach as many people as possible. Leave your questions in the comments below. I read them and I do my best to respond. And if you have not already subscribed to this channel, do that now, because we are just getting started. Thank you for watching, and I will see you in the next one.